Hello everyone, welcome to the class. Today we are going to discuss about population ecology, the basics of population ecology. Before going into population ecology, let us first clear the term population. What is population? So if we talk about population, it is basically the collection of an individuals of the same species that live together in a region. Suppose this is an area, and in this area there is only the birds, only the bird population is residing, right? So this is what we call population. That means it is a collection of individual of the same species. Make sure of the same species that live together in a region. We call it is a bird population. Similarly, if in a particular area there is a group of monkeys, then we call it's a monkeys population, right? So it is basically the collection of individuals of the same species that live together in a region, right? Now if we talk about population ecology, it is basically study of populations, their abundance, and how they change over time. So if we talk about this area, suppose it is say this area is rich in human population, right? So in case of population ecology, we see how this population of humans get changed over time. We see about the population abundance, we see about population abundance, we also see about pattern of abundance. Yes, pattern of abundance, how organism, how this species or how these humans changes over time, right? So that is what we call the population ecology. Coming to parameters to measure population. So how can we measure a population in a particular area? So to measure the population in a particular area, we have some sort of parameters like population density, mortality. Population density is basically the size of the population. Size of the population. So we have some process to determine the size of the population in a particular area. Then mortality. Mortality is basically what? It is a death rate. Death rate, right? What about natality? Natality is your birth rate. Birth rate. Then come your distribution. Distribution is basically your pattern. How the population is distributed in a area, right? We call it what? We call it distribution, right? So these parameters we need to check to measure the, just hold on. So these parameters we need to check to measure the population in a particular area. Now coming to population density. So if we talk about population density, this is how we define population density. This is basically the size of population, the size of a population residing a particular area. We represent it with the help of the population density, right? So now coming to the term density. Density, if we talk about population density, it represents the size of the population in a particular area, population of species in a particular area, right? Now, if we talk density, if we define the term density, what is density? It is basically the total number of individuals per unit area or volume at a given time. So suppose this at a particular time, how many individuals at a particular area, how many individuals are present? That thing we represent it with a term called density, right? So in case of population, we basically measure, we find out two types of density. One is specific or ecological density, another one is your crude density. Now what is ecological density and crude density? If we talk about ecological density, that is basically density per unit of habitat space. Suppose this is an area here. In this particular area, and say in this site only the birds are served, only in this particular area there is, you see, there is a population of birds, but this area there is no any bird present. So when we study specifically this particular area where a bird is present, where the group of birds are present, we call it a specific or ecological density, right? In case of crude density, we measure the entire area in case of crude density. But in case of ecological density, only that particular area where the organism is residing. We call it a specific density or ecological density. In case of crude density, it is overall density per unit total space. We calculate the total space. We don't We are not very specific in case of crude density, but specific or ecological density is very specific. It only measures that particular area which an individual occupies, right? Now, next we need to see the process how to determine population density, how population density is being determined. So to determine the population density, there is a process, we call it mark recapture technique. So mark recapture technique is basically, as the name suggests, it basically deals with 4 steps. What are the 4 steps? You can see in the name itself, mark and recapture. That means if this is an area here, and say there is some population of say insects present here, so what it will do, in case of mark recapture, there will be a capturing of the individuals, capturing of living organisms randomly. First step in case of mark recapture technique, what we do, we capture living organisms randomly from a particular area, right? Once the organism is captured, we mark them, we put some tag over the organism, right? Then what we do, we release those organisms in the same area, right? So first what we did, we captured the organism from the particular area, we marked the organism whatever we have captured, then we have released them in that particular area, right? Now again, what we do, again we recapture the organism from this area, and during this recapture, it is not confined, it is not confirmed that only the marked individual which we have captured before will come. There may be some other uh, organism which is not yet marked, right? So what we do, basically in first step in mark recapture technique, we randomly capture the, say this is a area, so I have defined with a particular example here, this is an area, in this area there is a population of birds are present, right? Birds, a group of birds are present, so we call it a bird population, right? So what we do, we randomly capture birds from this area, right? Then we tagged or marked the individual with some bands or with some color, right? Then we released it in that area, so that released bird will be mixed again, then again we recapture randomly. So here you need to know one term, whatever the organisms we have captured randomly, the first step, the organism what we get will come under sample 1, right? And whatever the individuals we marked, we represent it with mark individuals, released, and then again when we recapture it is considered as sample 2. When you recaptured again the organism, it is considered as sample 2, and in this sample 2, what happened, what is the chances that the individuals are marked or unmarked? There is a two-way chance, means either individual will be marked or unmarked. So sample 2 is a mixture of both marked and unmarked individual, right? So there are some numericals we'll be getting from this part, we need to see this, okay? So what we have studied now in mark recapture technique, we have seen that in a particular area, if a group of organism is present, we capture one organism, we marked it, how we marked, we put some tag, right? Then we released them, and again we recapture the organism. So first randomly captured organisms, we put it under sample 1, right? Then we marked the individual, marked will be considered as marked or tagged individual, which will be represented by M. Third, released, there will be no representation for released. Then again when we recapture, it will be considered as sample 2, and that sample 2 has a chance of having both marked individual and unmarked individual, right? So we'll move to a formula. So here this is the formula. You can see here N is basically the population size, right? S1 is the sample 1, S2 is sample 2, it can be marked or unmarked, right? And here this is M stands for marked individual. Suppose in the previous example, suppose this is an area, okay, and here I have seen there are some butterfly species present, okay? So what I have done, I have captured randomly, say I have captured randomly 50 butterflies from this sample, right? Now out of 50, I have marked 30, or I have marked 50 individuals. I have captured in the sample 1, I have captured 50 individuals. I have marked all the 50 individuals and then released. And again in recaptured, what I got, I have captured say 150 individuals. Now what is the population size? So in this case, what will be the population size? S1 into S2 divided by M. So what is the S1? It is 50. S2 is 150. And number of marked individual is 50. So you just calculate. It is equal to 150 is the answer. Clear? So we see some questions from this part. Suppose here is another question. Here it states that an individual took 200 birds. So here 200 birds will be your sample 1. Right? Sample 1 is your 200 birds out of a forest. Put leg bands on them, return them to the forest. If the individual then take 250 birds, if the individual then take 250 birds, so here sample 2 is equals to 250, right? From the forest, and find 50 of them to have leg bends. Leg bends is what? Kind of marked, right? So here M will be your 50. Then what is the population size? Population size will be how we will represent the population size? We have the formula N equal to S1 into S2 divided by M. So here S1 is 200, S2 is 250 and marked is 50. So you can see we have 1000 population size. So population size is how much? It is 1000, right? So let's move to another numerical here. So here also, to determine the rats in the field, 80 rats were initially captured. So that will be your sample 1, right? 80 will be your sample 1 because that was initially captured, marked and released. After 1 month, 100 rats were captured. Again, 100 rats were captured. So that will be your sample 2, right? In the same field, of which 20 were previously marked. So M is here 20, right? So based on above observation, estimated population size of the rats in the field will be. So again, same formula, N is equals to S1 into S2 by M. So here what we got in the first capturing, we got 80 rats. So here S1 sample 1 will be your 80. Right? And in case of sample 2, when he recaptured, he got 100 rats. So that will be your sample 2. Then out of 100 rats, 20 are marked. So here you put it in the formula 80 into 100 by 20, you will get 400. Right? So that is all about today's class. Today we have seen about the basics of population ecology. Next class we will be dealing in detail about the graphs, about the population dispersion pattern, everything we will study. Right? Thank you so much students for joining the class.